Good afternoon, everybody. I am Dr. Erica Ollmann Saphire, President and CEO of the La Jolla Institute for Immunology, or LJI, as we like to call it. It's a real pleasure to welcome you to this session of our Live from the Lab series. Today's webinar, entitled You, Me, and Measles, hits especially close to home for me. Not only does it highlight the groundbreaking work happening right here in my own laboratory, where I'm a professor, but it also underscores how our community of supporters is the driving force behind these scientific discoveries that propel our mission forward. So imagine, if you will, imagine a virus so cunning, it would invade your cells like a master thief, if not for an army of antibodies intercepting it before it ever gets the chance. My team in my lab, using cutting-edge cryo-electron microscopy, unveiled the first structure of any antibody to halt the measles virus, right as it's about to fuse with its target cell. So that breakthrough not only paves the way for novel measles treatments, the first of their kind, medicine doesn't have any yet, but it also holds promise for tackling other formidable family viruses in the whole family, of which measles is just one member. We were thrilled to recently share this collaborative research with the scientific community in the prestigious journal Science. But before we show you what those incredible images look like, I want to take a moment to extend my deepest gratitude to a few key individuals whose generosity has helped make this research we're discussing today possible. First, a huge thank you to Michael and Elise Coit. Their generous funding of the Beacon equipment has been nothing short of pivotal in advancing our research. You will actually get to see the Beacon in action live in this webinar today. Its name comes from the fact that it shines a light forward. Quite literally, in this instrument, we find rare B cells that we need, and we can find them because they function like tiny little emitters of beacons of light, and that helps us identify them in the pool. And we can actually, again, use light itself to lift those B cells out and grow them for further analysis. And I also want to shine a light on the significant donors who made it possible for us to acquire our Titan Krios cryo-electron microscope. Their and others' contributions have been transformative in the truest sense of the word. In particular, I'd like to thank board directors Tony Carr and Gail Naughton, board chair emeritus John Major and his wife Sue, the Overton family, the GHR Foundation, and the Bill and Melinda Gates Foundation. Another special thanks goes to John and Sue Major, whose support was instrumental in creating the John and Susan Major Center for Clinical Investigation. This center is the beating heart of our research, providing the crucial clinical samples that fuel the discoveries we'll explore today. And last but certainly not least, thank you to Board Director Barbara Donnell, Susan and David Budd, as well as Maria Silva and Bill Passy. Their support through the Tully and Ricky Family Spark Awards program allowed Dr. Dawid Zyla to lay a portion of the groundwork for the research we'll be highlighting today. Our featured researchers today are Dr. Zyla and Dr. Marissa Acciani, and their combined expertise is pivotal in our quest against measles virus. Dr. Zyla will share his latest research on the structural intricacies of the measles virus and the immune response against it. His work is fundamental, and it gave science the blueprints for the molecular machines that allow measles virus to drive itself into your cells, as well as the maps of what antibodies we can use to develop into targeted therapeutics. Think of Dawid as the cartographer of this scientific expedition, charting out the landscape that will guide future interventions. Following Dr. Zyla's exploration, Dr. Acciani will provide an exclusive behind-the-scenes look at one of our lab's technological marvels, that Beacon instrument in the Coit Antibody Discovery Center. That advanced tool is not just a piece of equipment, it's a game changer that allows us to find those rare antibodies that can stop viruses in their tracks. Dr. Jolla's structural insights, combined with Dr. Acciani's expertise in antibody mechanisms, bridge the gap between understanding the virus and applying that knowledge in practice. Today's session will showcase how their collaboration drives breakthroughs, with Dr. Jolla laying the foundation and Dr. Acciani translating it into actionable solutions. Their work exemplifies the transformative power of cross-disciplinary collaboration, breaking down silos to tackle and solve complex disease challenges. This caliber of collaboration is what sets LJI apart from its peers in the medical research community. After hearing from Dr. Jolla and Dr. Acciani, we will open the floor for a Q&A session. And throughout the presentation, I encourage you to submit your questions via the Q&A function in Zoom at the bottom of the window. We'll do our best to get to as many as we can. So thank you all for being here today. Your support is what enables us to push the boundaries of science and to do it together. Let's get started. First, I'll provide a very brief introduction of LJI for those who are new to the Institute. So let me share my slides with you, and I will push play, and I will go back to the beginning. So we work at LJI on the human immune system. And there's a reason we focus on the human immune system, is that nothing else has the singular power to cure or prevent and sometimes cause nearly every human disease. You grow up knowing that the immune system could defend against infectious diseases. But it turns out the immune system also plays a critical role in heart disease, in cognitive health and neurodegenerative disease, in finding and destroying cancer cells that have escaped any surgeon's scalpel. And of course, it is the critical agent and cause behind autoimmune disease, with cases growing every decade, especially in girls and women. So these diseases that strike our families, like cancer or heart attacks or autoimmune disorders or so many other things that are complex and as yet not fully understood, they're taking a heavy toll on us and our families every day. And it's more urgent than ever to now understand the differences between people. Well, some get sick and some do not. It's not a mystery. We can put together the piece of data that explain the mechanism by which disease arose and what intervention we need to put in place to cure it and prevent it. We're also understanding fundamental differences in the immune responses between women and men that offer a wellspring of innovation and information that help everybody. And we're developing better vaccines to understand why some confer lifelong immunity and others do not. We are a nonprofit private research institute. So we are by scientists for scientists. We were founded about 35 years ago, but from this little jewel in La Jolla with, 21 laboratories, 451 total staff, and 181 postdoctoral fellows and other trainees. From this little gem, we are ranked top 5 in the world for the fundamental study of the human immune response. It's also an extraordinary place to work with a terrific culture, and we've won Best Workplace in San Diego every year for the last 10 years running. I also want to introduce to you the 2 technical marvels, 2 among many here in our institute, but these are the 2 we'll be talking about today. So on the left, the cryo-EM, cryo-electron microscopy facility at LJI. It is a transformative force in structural biology. It uses advanced technology and an exceptionally powerful electron beam to capture high-resolution images of biological macromolecules in action. It illuminates biology at the molecular nature in its most authentic state. By freezing the samples and illuminating them with an incredibly powerful electron beam, we can explore molecular structures with unparalleled clarity, directly seeing, directly visualizing, and understanding how disease unfolds and how the immune system defends. On the right, the Beacon instrument. That platform in our Coit Antibody Discovery Center, it's another technological marvel. And it has for us revolutionized the search for rare antibodies with unparalleled precision and speed. By isolating elusive B cells that produce the best virus-blocking antibodies. That instrument you're looking at right there accelerates the development of potential treatments. That cutting-edge technology turned possibility into reality for us at LJI and for all the scientists around the world with whom we collaborate. Now, you will learn more about both the cryo-EM and the beacon shortly. It is my pleasure now to formally introduce the two speakers for today, Dr. Dawid Zyla and Dr. Acciani. First, Dr. Marissa Acciani earned her Ph.D. from the University of Georgia in Athens, where she laid the groundwork for her research. Since joining my laboratory here, she has focused on characterizing the antibody responses triggered by the measles virus vaccine, using blood samples donated in our clinical core by local San Diegans. Her postdoctoral research has expanded our insights into the immune system's reaction to a range of viral challenges. And I'm also pleased to introduce Dr. Dawid Zyla. Born in Poland, Dawid carved his academic path in Switzerland, where he immersed himself in the intricate world of protein structures central to viral infections. Now, as an instructor in my lab, he's taking a close look at measles, dissecting how this virus maneuvers through our immune system. Dr. Zyla is also an exceptional photographer, and it was his own work that was used for the invitations to this webinar. So with that, Dawid, take it away.
Hello, everyone. Good afternoon. I will just share my presentation now. All right. So as Erica mentioned, my name is Dawid Zyla, and I'm interested in discovering how proteins refold and how their change in structure drives the action of the proteins. And in this case, I want to show you our work in the lab of Erica. But let's start first with the question, what are even viruses? Viruses are very small. They are roughly 200 nanometer in size, and they are very hard to imagine when you think about it. They are way smaller than the bacteria that are also pathogenic, and they are tiny in comparison to our cells. When we zoom in on viral particle, we can see that they are made of the genetic material, which can be either DNA or RNA, and they are surrounded by the protein coat with sometimes also a lipid layer. However, viruses are not considered alive on their own. They cannot grow, they cannot move, they cannot reproduce by themselves. Instead, they need to infect a living cells and use the cell machinery to make more copies of themselves. But why the viruses are important? I think this question is relatively simple. Viruses are everywhere. They are in the air we breathe, they are on the surfaces we touch, they are even inside our bodies. Despite being tiny, they have an enormous impact on our lives. They can evolve quickly, which is why new viruses appear, and this is why it's very hard to control them. But how we can protect ourselves from viruses? The first are vaccines, and they are one of the most powerful tools we have to prevent viral infections. They train our immune system to recognize and fight viruses before we even come into contact with them. Vaccination doesn't just protect a person receiving the vaccine, it also helps to generate a population-based shield called herd immunity that protects vulnerable people who cannot get vaccinated, like babies or those with weakened immune system. In addition to vaccines, practicing healthy habits like regular hand washing, wearing masks during outbreaks, and avoiding areas where the viral infections are spreading can help reduce our chances of being exposed to viruses. These are simple ways to protect ourselves and others. And what is also very interesting, that they were super successful during COVID-19 pandemic. Even if someone gets infected with the virus, there are some treatments available that can help. For center viruses, we have small molecule drugs and antibodies that can reduce the severity of the illness. However, unlike antibiotics for bacterial infections, we have far fewer antiviral drugs. Most of these treatments help manage the symptoms rather than completely cure the infection. For example, for COVID-19, we have now Paxlovid, which helps to reduce the risk of severe illness. Also, for the cold sores, we have acyclovir, which helps to manage the severity of the disease. But overall, the antivirals are very limited, and they often rely on the treatment relies on the support of the recovery rather than eliminating the virus entirely. Finally, there is research. So the stuff what we do here at the Institute. We study viruses and understand how they spread, how they infect cells. This research is essential for developing the vaccines and new treatments, and it helps us to understand how viruses work and how we can prevent viral future threats. I also would like to focus on this part today and give you a short overview of our work and how we contribute to life without disease. Among many viruses we study in the lab, we focus today on measles, as Erica mentioned. Measles is an ancient virus, and it's one of the most, actually, it's the most infectious disease we know. This means it can spread incredibly easily from one person to the other. In fact, just being in the same room with someone who has measles can result in infection even hours after they left, and a single person can pass it to 16 to 18 unvaccinated individuals. To protect everyone, especially those who cannot get vaccinated, we have to have at least 95% of population to be vaccinated. This high level of immunity creates what we call herd immunity, where the virus can spread easily because most people are protected. And the vaccine, it cannot drop below this threshold because the outbreak can really become and spread. Despite being preventable, measles continues to cause more deaths in children than any other vaccine-preventable disease. It's particularly dangerous for young children, leading to complications like pneumonia and brain swelling, which might be fatal. Measles is a high-risk threat in India and many African countries, where even 40,000 cases were reported already this year. Recently, our efforts to vaccinate against measles have been compromised. The COVID-19 pandemic disrupted many vaccination campaigns, making it harder to keep measles under control. Additionally, misinformation and sociological factors like vaccine hesitancy have reduced vaccination rates in the US and globally. It is important to remember that some people, like I mentioned, the newborns and the people with certain medical conditions, they cannot get vaccinated, and they rely on the rest of us and the herd immunity. This is why it's so important that we achieve high vaccination coverage so that everyone is protected, even the most vulnerable ones. Measles once was believed to be eradicated in the US in early 2000. However, the number of cases has increased since then. As you can see here on this plot, where I will have the pointer, on this plot here, many US states, they dropped below this 95% herd immunity threshold, and they risk raising measles outbreaks. Some outbreaks already happened in US, like the one in 2019 in New York and last year in Ohio. Fortunately, we have a vaccine for measles, which is one of the most effective vaccines ever developed. Since the introduction in the 1960s, it has saved millions of lives by dramatically reducing the number of measles cases worldwide. It's been over 95% effective in preventing measles in fully vaccinated people, making it one of the best tools we have to fight this highly contagious virus. The current measles vaccine contains a live, attenuated, which means weakened version of the virus. This weakened virus can't cause illness, but it's strong enough to stimulate our immune system to respond and produce antibodies. Once a person gets vaccinated, the immune system starts to work. First, the naive B cells in our immune system recognize attenuated virus and see it as a threat. In response, those B cells begin to produce antibodies, proteins they are designed to fight the virus. Those antibodies will attach to the virus and stop it from causing infection. At the same time, the immune system creates memory cells. These memory cells stay in our body for years, keeping the blueprint of the measles virus. As you can encounter the real virus later, those memory cells will recognize it immediately and quickly produce antibodies to prevent from getting sick. While the vaccine is incredibly effective, there are some limitations. Some people, like infants, which are too young to be vaccinated, or people with weakened immune systems, they cannot receive this vaccine. Additionally, in rare cases, some people might not develop complete immunity even after being vaccinated. But for most people, this vaccine works really well. However, people still get infected with measles, and recently the cases are spiking. So the question is, how many post-treatment and measles-specific treatments and drugs do we have? We have none. And developing new antiviral strategies for measles is really essential. While the vaccine has been incredibly successful, we still do not fully understand why this vaccine works so well. And the research on measles is crucial to answer those questions. By studying the virus and how it interacts with our immune system, we can improve the treatments and protect those who cannot be vaccinated. Research gives us tools so we can stay ahead of the virus and prevent future outbreaks. Before we begin and develop new treatments, we need to understand how the virus works and how it looks like. On the surface of the virus, there are two specialized proteins. In green, fusion glycoprotein, and in purple, the attachment glycoprotein called H or hemagglutinin. Our cells, they have a series of receptors or the surface proteins that are recognized by the measles virus. Those receptors are recognized by the attachment protein, and the attachment protein tightly binds the host cell receptor, anchoring the virus on the host cell membrane. This is a critical step which allows the virus to prepare for the membrane fusion. After successful receptor binding, a conformational change happens in F, known as F triggering. This leads to the F protein unfolding and forming a pre-harping intermediate state. This structure extends towards the host cell membrane, allowing the viral and the host cell membrane to come close. As the F continues to rearrange, it pulls the viral and host cell membranes even closer. The extended harping intermediate structure collapses, driving the fusion of those two membranes. And finally, the viral and host cell membranes merge or fuse, allowing the viral RNA to enter the host cell. Once inside, the viral RNA can hijack the host cell machinery to begin the process of replication and production of the new viruses. As the fusion glycoprotein, or F, plays a central role in the membrane fusion process, we decided to focus on this protein for our studies. One of the virus's main challenge is actually entering host cells. By investigating how F protein works during viral entry, we aim to develop potential treatments which use the vulnerabilities present in this system. But how we can even study viral glycoproteins that are even 100 times smaller than the virus itself? The answer is, as Erica mentioned, is cryogenic electron microscopy, or cryo-EM. This powerful technology allows us to see tiny particles at the magnifications over 100,000 times using microscopes that are over 10 feet tall and cost millions of dollars. To put it in the perspective, if you would magnify a baseball to this level, it would have almost 5 miles in diameter. Our work to image proteins begins in the lab, where we produce and purify viral glycoproteins. We then quickly freeze these proteins on the grids, those copper rings that are 2 millimeters in size, using this device shown here, which generates a very thin layer of water on those grids and freezes them in liquid nitrogen temperatures. We have those tiny small copper or gold rings where we put our protein on, and then we use this device to generate a very thin layer of water and then freeze it. Then those grids, they are loaded into this microscope shown here. And then when we load them on the microscope at the low magnifications, we see this pattern here. In this pattern, we can see the white and the dark spots, which corresponds to the protein regions and thick ice regions. We can use this microscope to go even further and zoom in. So now in every of those small white spots, we have this pattern of holes. And in every single hole here, we have multiple protein copies frozen. But to see it, we have to go even further to 130 times magnification. Here, finally, you can see the individual copies of the protein, but let's make this image slightly bigger. So now we have here a single cryo-EM image, which contains hundreds of copies of measles glycoprotein. Each black blob here represents a single protein, which is less than 15 nanometer in size. When we zoom in even further, we can see a single protein here with a single glycosylation site. But this image is unfortunately very blurry and it has a lot of noise. To improve the quality of the data, we need to record thousands of images like this one here and extract from this image hundreds of thousands of the particle images shown here. And the final step uses advanced computational analysis to build detailed map of the protein structure, allowing us to assemble the structure and see how the protein exactly works. So we used cryo-EM to capture high-resolution structures of measles fusion glycoprotein in those two important steps, which is the prefusion state and the post-fusion state. These were the first ever cryo-EM structures of measles glycoproteins resolved to show those states. And also this was the first post-fusion state of measles glycoprotein. With both the starting and the ending point of the fusion process in hand, we could model how those glycoproteins changes during the fusion. So we built the atomic models into them. And here you can see that we have labeled each individual domain of the protein in the prefusion and post-fusion state. And now we can iterate between those two states. So now we can see the animation that iterates from between this state and this state here. And the compact prefusion state opens up, launching towards the host membrane. This process eventually brings the viral and host membrane together, allowing the virus to enter the cell. It is remarkable how much change happens in this protein so the virus can infect. So now we understand how the fusion protein refolds. Our next goal was to find the way to stop this process, which would also prevent the measles virus from entering our cells. To do this, we decided to investigate the only known antibody against measles called monoclonal antibody 77 or MAP77, which was discovered in 1990s and also recently described by our collaborators at the Columbia University. We compared the effectiveness of this antibody to two previously described inhibitors for measles fusion glycoprotein. While neither of those two small molecules has been approved for treatment in humans, they serve as a valuable tools for studying the fusion process. We found that this antibody is much more effective in stopping the virus at much lower concentration in comparison to the previous two inhibitors. Interestingly, we also know how those inhibitors work. Particles of the glycoprotein bound by the antibody. On the right, we have the classes which were collected from the same particles. And this gives us the image where the antibody binds on this glycoprotein. So the particles shown here again on the left, they are used to generate the 3D structures and build the atomic models, which here on the right is shown in the ribbon. What we can see here is the first structure of measles fusion protein
Hey, folks, it looks like we've dropped our connection. So while we try to get Dawid back on, let's throw it right to Marissa in the Coit antibody discovery. Hi, Marissa.
Hi,
So
Erica.
he's going to show us the behind the scenes look inside the lab and that super special beacon instrument that lets us illuminate the B cells that we want to study.
Thank you, Erica. So as Erica mentioned, and as Dawid will discuss when we get back to him, vaccines and the protective antibodies that they generate are critical for fighting infectious virus in the body. And purifying and studying those antibodies helps us understand our incredibly complex immune systems and the pathogens they target. This is the purpose of the Saphire Labs Antibody Discovery Center, which was established with the support of Michael and Elise Coit, and through the hard work of Dr. Catherine Hasty, Stephanie Harkins, and Michelle Zandonati. Let's check it out. So our antibody discovery pipeline begins here with the Beacon optifluidic system. Now the Beacon is unsurpassed in terms of analyzing thousands and thousands of cells individually rapidly. And I'll explain why that's so important to my project. So when we're vaccinated or infected with a virus, we generate vast populations of plasma B cells that release antivirus antibodies into the blood. Each cell produces a unique population of antibodies with distinct abilities to bind to and stop the virus. Now, we can learn some things about mixed populations of antibodies, but we can learn far more by studying these antibody populations individually. And this is where the Beacon comes in. So we can inject tens of thousands of our plasma B cells onto the Beacon, which will separate them and tell us which ones generate antibodies against our viral protein targets, which in this case are the measles virus surface proteins H and F, or hemagglutinin or fusion. Then the Beacon will export the DNA from each cell, which we can then use to clone each antibody and produce it ourselves on a much larger scale for further analysis. So for my project, I'm studying the memory immune response to the measles virus vaccine. For that, we start with memory B cells from vaccinated donors who donated their blood to our clinical core. Then we transform those cells into plasma B cells. Those are the ones that release the antibodies in the lab. Then we can inject those cells onto our Beacon chip for individual cell analysis. So here I have an example of our Beacon chip. This chip contains 11,000 nanopen chambers that will house our individual cells. Here is our reagent bay. So that will contain the cell media that will be responsible for keeping our cells alive and happy and healthy throughout the experiment. Now here in my pipette tip, I have 20,000 memory B cells transformed into plasma cells from a measles virus vaccine. I'm going to inject these cells into this inlet here, close the door, and the Beacon is going to transport these cells into channels that flow above our nanopen chambers. And I can show you a video of what that looks like. So these cells are incredibly small, so hopefully you can see them, but they are perfectly round, little dots. Here's one, and here's one. So they'll pass through the channels above the nanopen chambers. Then the Beacon is going to identify the single cells, so you'll be able to see them better. It puts a box around these individual cells and uses light to transport each cell into its nanopen chamber. Down into the chamber. So next, we inject our measles bait, which are measles H and F proteins that are labeled with fluorochromes that emit different color lights, which are detectable by the Beacon. Those probes will also pass through the channels above the pens. If our plasma cells are releasing antibodies that can bind to those probes, we'll see a fluorescent bloom of signal above that pen. And I can show you an example of that here. We'll shut off the lights. So these are plasma cells that are generating antibodies against our measles H and F proteins. And I can play that again and pause if anybody has a hard time seeing those signals. So here, for example, is a pen containing a cell that is producing antibodies that can bind to our measles F protein. So once our experiment is complete, we can tell the beacon to lyse our cells of interest and then export the DNA from each cell into a well on our microplate. We can then sequence the antibody genes from each of our cells, produce the antibodies ourselves, and use them for further analysis. So for example, we will determine where on our H and F proteins the antibodies bind and the strength of those interactions. Our ultimate goal will be to determine which sites on our H and F proteins are most targeted by antibodies produced when we are vaccinated with the measles vaccine. Oh, yes, and the mechanisms that those antibodies use to block those interactions. And Dawid is going to explain with an antibody called 77 what kind of experiments you can do to figure out the mechanism of action of those antibodies. When he froze, he was just in the middle of an experiment like that. Ultimately, we can learn so much from one single antibody, which he'll explain. But imagine what we could learn with 10, 20, 50, 100 antibodies that bind to our measles H and F protein. Thank you, guys. And with that, I'll pass you back.
Hello again. Sorry for the technical difficulties. Okay. All right. So hopefully this was the last slide which was going through. So as I mentioned, we know now how the fusion glycoprotein works, and we wanted to actually stop it from working, which we can use and which we can exploit this mechanism to maybe develop new antiviral strategies. So we use the antibody from described recently by our collaborators, and we discovered that this antibody works much better than previously characterized small inhibitors against measles fusion protein. While neither of these two small molecules has been approved for treatments in humans, they serve as valuable tools for studying the fusion process. We found that our antibody is much more effective in stopping virus infection than the previously characterized two small inhibitors, and it works at much lower concentrations. We also knew how the mechanism of those small inhibitors looks like. So one of the inhibitors blocks the fusion protein in the prefusion state and doesn't allow it to refold, while the second one locks the intermediate state and it doesn't allow to go through the collapse and membrane fusion. So we asked the question, how the antibody 77 exactly works. So we came back to cryo-EM and formed complexes between the fusion protein and the antibody fragments. And then on the left, you can see the antibody bound glycoprotein. And on the right, there are 2D classes which are coming from the same complex. And this data we can use to generate a 3D structure. So here, those images, they show different positions of the particle in ice. And on the right, we can see the final cryo-EM map. We've modeled our structure in it, shown in ribbon. So in the middle, we have the fusion glycoprotein in green. And then on the outside, we have those antibody fragments binding on the bottom side of the protein. And this whole structure is similar, like a triskelion shape. So we notice that the antibody binds at the interface where 3 chains of the protein meet, and this antibody might be stabilizing the perfusion state. With the cryo-EM, we were able to identify where the antibody binds to the fusion protein, but we still didn't know how this antibody stops the virus. Fusion is a dynamic process, and the antibody must block one of the key steps. And our collaborators have developed a clever cell-based assay that helps us to determine at which step of the fusion this antibody works. Using this assay, we can test a few possibilities. So the first one is where the antibody might block the activation of the fusion protein, keeping it locked in the prefusion state. And the fusion protein cannot progress to the next step. And because the host cell is just connected through the receptor, we call the state reversibly bound. In the second possibility, the fusion protein was activated and launched towards the host cell. However, the antibody would block the fusion from progressing, and the fusion wouldn't finally happen. In the final scenario, in the absence of the inhibitor or antibody, the virus fuses normally and the genetic material can enter the host cell. In the control experiment, without any inhibitors, we observed that most of the cells successfully fused, while only few remained in the reversibly bound state. When we introduce one of the inhibitors called 3G, it almost completely blocks the activation of the fusion protein, keeping it locked in the prefusion state here. Another inhibitor called HRC4 binds to the fusion protein after it has been activated and initiated contact with the host membranes. However, this inhibitor prevents the fusion process from completing. In this state, the protein stays in the intermediate state and it stops the fusion machinery from progressing. So finally, we have the mechanism of antibody 77. Surprisingly, it works a very similar way as HRC4, blocking the fusion after the protein has started refolding. This result was puzzling because our cryo-EM data suggests that the antibody actually recognizes prefusion state. So because of this confusion, we went back to our cryo-EM data and reinvestigated whether there are any clues that we have missed, and we could explain how the antibody works. So we took a closer look at our cryo-EM data set again, and we discovered that the antibody binds not just the prefusion state, but also intermediate stages of the protein when it starts refolding. This helped us to understand how the antibody works and blocks the virus. At the top, you can see the raw images or the 2D classes from the microscope, and at the bottom, we have the annotated data. So in the first image, the antibody is bound to the protein the same way as I was showing you before, forming this triskelion-like shape. In the second state, we notice a small orange part, which previously was shaped like this, forms a spherical shape in the needle, which shows that the protein refolding has initiated. In the first state, the protein begins to come apart with only two parts of the protein still connected. Finally, at the last state, we see that there is just the antibody fragment binding a small fragment of the fusion protein. So what we have discovered is that this antibody breaks the protein as the protein starts to refold. This data was very exciting as we have not only characterized a new antibody against measles virus, but also showed a novel mechanism by which neutralizing antibodies can stop viral infections. We also plan to build on this information and also study other antibodies. So we have characterized only a single antibody called MAP77 against measles, and story of this antibody goes back to 1990 when it was discovered, and it was discovered through vaccinating mice. But this is where the exciting part is. We've been all vaccinated against measles, which means that we all have antibodies against this virus. So imagine how much more we can learn by studying the antibodies produced by this vaccine. It could help us understand why the current vaccine is so effective and how we can use this knowledge to improve other vaccines. So this is where Marissa's project begins, and she already guided you through the beacon. I will just very quickly show you the schematic. So first, we isolate the memory B cells from the MMR vaccine. The blood was collected in our donor center here at the LJI. Then we use, as Marissa mentioned, we separate the cells which are specific to the measles glycoproteins, and then we transform those cells from memory to plasma cells which are actively producing antibodies. Then, as Marissa showed, we have this nanochip, which has thousands of small pens where the individual cell can sit. Then we use the beacon device, as you could see it at the presentation. And then we can put the cells inside the pens, and then we can investigate where those blooms, as Marissa mentioned, happen. Then we lyse, or we open up the cells, and we select only the ones which gave us the positive response for the measles antibodies, and we generate the DNA out of the mRNA, and then we can produce, based on this information, the antibodies, which we can use for many other stuff. But the question is, like, why do we even need the antibodies for? So antibodies are incredibly useful tools for understanding how protein work. For example, antibodies like MAP77 can bind to the protein and stop this at different refolding stages, helping us to study the protein's function and refolding timing. But antibodies are much more than just helping us in the lab. They can also be used in the therapeutic cocktails to treat active infections. We've seen this approach to work successfully for treatments for diseases like Ebola and COVID-19, where antibody therapies have helped patients recover faster. Lastly, antibodies can be used as a diagnostic tools. They can help us to detect viruses in water, food, or even our blood, allowing us to identify infections early and improve their prevention efforts. So as I've shown you today, and Marissa, that the basic research we do here in the lab at the LJI help us to understand how the viruses infect and how we can stop them. We also highlighted the critical role of generating new antibodies, which provide us with a valuable research tools, innovative treatments, and improved diagnostics. The information we gathered from studying measles, which serves as a model organism for the whole family of the viruses where measles is a model organism, can be directly applied to other dangerous viruses like Nipah and Hendra, as well as other viruses that pose threat in the future. The research we conduct here in the lab not only help us fighting the current viral threats, but also prepares us for the viral challenges of tomorrow. So this brings me to the end of my presentation. And I would like to thank everyone who has contributed to this project, and special thanks to Erica, Marissa, and Gayla, and all the members of the Saphire Lab for helping with this project. I also would like to thank our collaborators in New York at Columbia University, Matteo and Anne, and our collaborators at Utrecht and Ohio State University, Just and Stefan. None of this cutting-edge research would be possible without the generous support from NIH and Swiss National Foundation, and also from our very generous donors who support cryo-EM and the Beacon. And thank you for your attention.
Thank you so much, Dawid. And Marissa has rejoined us, come in from the lab. So now is the Q&A. And I've got a couple of questions from the audience, but please feel free to put more into the chat. We'll start with one thing that I get a lot, which is, humanity has known about measles for hundreds of years. Why are you guys the first to give us a structure of what the thing in the immune system looks like? Why are we only just seeing these things now?
It's very difficult to study responses from the vaccine while we do not have good proteins. And I think the biggest achievements here from us and also from the Columbia was that we have really good proteins now that we can use to study the virus, and we can study the antibodies. Without the baits, as Marissa mentioned, we couldn't get the antibodies against measles.
Yeah. So another question is, so Marissa's looking at antibodies, then the array of antibodies from people that got the measles virus vaccine. And we also have other people in our clinical core that had a measles virus infection. But what some of our people say is they come in and they say, well, I got the first and second doses of measles vaccine as a child, But sometimes they've done some recent blood work in preparation for something else, and the doctor noticed that they don't have detectable antibody titers against measles, either anymore or at all. So how can that be? Why does immunity for some people diminish over time?
I'll take this. So everybody who receives a vaccine will have an individual immune response to that vaccine. So we know for the measles vaccine, the 2 doses, that generally immunity will be lifelong and will give you lifelong protection and it will be durable. But there are going to be some people where your immunity will wane over time. So your population of memory B cells with specific antibodies that target the virus, those populations will start to wane over time, which means if you get infected, you might develop disease, but your risk is incredibly low.
And then I guess the other good analogy is that your immune system wants to, it's a lot of caloric output to make an antibody response against something. And if it's a virus your immune system hasn't seen in a while, thinks I might not need to keep stocking those shelves. Like Target doesn't keep the snow boots and the mittens on the shelves in January because there's no demand for them. But if people started coming in and asking for them, they'd pull them out of storage and put them back on. Yeah, July. So that gives us almost to the point where most of us have never come across measles because of the historical vaccination campaign, but failures in the vaccination campaign poises for an explosion. And there's a lot of people that are vulnerable or that cannot get the current measles vaccine because it's a replicating virus. So let's talk about the resurgence of the threat. So Dawid and Marissa, here's another audience question. At one point in time, we thought the measles virus was very nearly eradicated. We were ready to call victory, at least in the United States. But it's now come back with outbreaks in at least 14 different states. So how do you balance the current resurgence of the virus while also preparing for future viral threats of other viruses in the same family as measles?
So we are studying the fusion protein here. And what is really cool about this protein, that the mechanism, how it works the same for all the other viruses in the same family. We would not expect that the fusion protein of Nipah or paraflu-3 or Hendra or Newcastle disease virus would work much differently. So getting the blueprints of how this mechanism works, we can design maybe somewhere in the future treatment which can basically neutralize all the threats in this viral family at once.
And we know that viruses mutate and evolve. So how do we make sure that the vaccines and therapeutics being developed will stand the test of time?
This is a very good question. And this is also, we don't know how the evolution goes, but what we can assume is that the function of the protein on its own has to stay. So the virus cannot really mutate too much because this protein wouldn't do the function anymore. So the more we know about how the fusion process works, the more we can prepare for mutants which emerge.
Exactly. And then what about some of the recent advances like mRNA technology or gene editing with CRISPR? How might you one day engineer more durable immune responses to viruses like measles?
So understanding the most effective, potent targets that can apply to as many versions of your virus as possible is going to be critical. And then remind me of your question again.
How can you engineer more durable immune responses? And then, the correlating question is, what do you do for people that can't receive this kind of vaccine? Because even though it's a live replicating virus, a healthy immune system can knock it down. Like pediatric cancer patients can't receive even the weakened virus because the vaccine itself could be lethal. What do you do?
Sure. So improving your target, which was what I stated before, is definitely going to help so that whatever the amount of immune or memory cells that you generate, the antibodies that they're going to release when they encounter the pathogen, again, are going to be very effective in neutralizing your virus. Also, the current vaccine, you do need 2 doses, which automatically makes it more difficult to get the level of immunity that you need. So hopefully we can engineer a better vaccine that's more potent, potentially that only needs 1 dose, which will help with our overall coverage and our ability to establish herd immunity. And then it could also be safer for those who can't receive a live virus vaccine. So the vaccine that Dawid potentially has mentioned, have you mentioned new vaccines? Okay. So there are ways that you can vaccinate people with without live virus, with just very specific components of that virus that won't irritate your immune systems the way that a live virus will. So it will be safer to receive. Hopefully you can engineer alternative vaccines that are cheaper, that only require 1 dose, and will give you the same protection as the current vaccine, or even better, because you know the exact targets to hit.
And then the other thing is the antibodies themselves can be treatments. So a vaccine will protect you a month from now, but let's say you've already been infected and need treatment now. Those antibodies themselves are a first-in-class life-saving thing you can give someone who has come down with measles right away, and give them the immune response of a rock star the same day. So that's pretty remarkable. So what are the main challenges that you would face in translating treatments like those antibody treatments to clinical practice?
This is, I think, a returning problem for measles research, because we cannot, the only host of the measles virus is humans. So basically we would need to test everything on something which is very close to humans, which are non-human primates. So once we develop a treatment, we would need to test it so it's safe for everyone. And doing those tests, it poses some challenges.
True. All right, we are at the top of the hour. So I want to thank the audience, all of you, for joining us today for a live look inside the lab. Today, we have seen how the cutting edge research here in this building at LJI has been accelerated or propelled really by the remarkable generosity of our community. The breakthroughs you've heard about, from Dr. Jolla's structural insights to Dr. Acciani's pioneering antibody work, they don't just happen in isolation. They are made possible by the technologies that your contributions put directly into their hands. Those tools aren't just machines, they're the spark. They are the spark that ignites discovery, pushing us closer to the vaccines and the treatments that the world urgently needs. So if what you've seen today sparks something in you, if you want to be a part of this relentless drive to understand, heal, and protect, I encourage you to join us. There's a direct line between philanthropy and progress. And if you're curious how to get involved, LJI's Vice President of Advancement, Kelsey Dale, whose contact is on the slide, can tell you more. Our work at LJI is nowhere near done. We need the continued energy and support of people like you to keep pressing forward and keep taking on the impossible, things that had never been done for hundreds of years until they were done right here. Philanthropy is what allows us to chase those discoveries that aren't just ambitious, they are essential. Together, we can keep making strides against the global health challenges of our time. Before we wrap up, a quick reminder that we will be sending out a recording of today's webinar, so you can watch it again, or you can share it with your networks. Thank you for being here, for your time, your attention, and most of all, thank you for your belief in what we can do together. Have a wonderful rest of your day.